Hello everyone. So, in today's session of overview and integration of cellular metabolism, we will discuss TCA cycle, the biochemistry of TCA cycle. The concept here will be covered are conversion of pyruvate to acetyl coenzyme A and the different biochemical steps of TCA cycle. Now, before discussing TCA cycle, I would like to highlight that in the previous session we have seen that glucose is the major source of fuel and that can undergo aerobic glycolysis as well as anaerobic glycolysis. In mammalian cell, which is where the cells are basically aerobic mostly, there this pyruvate actually enters TCA cycle by forming acetyl coenzyme A, and then at the end of TCA cycle there is formation of few reducing equivalent which is utilized to form energy in the form of ATP in oxidative phosphorylation. Now, this is the basic concept of cellular respiration which occurs in three different phases. In cellular respiration, there are different metabolic fuels, mainly carbohydrate, lipid and proteins. Now, carbohydrate, lipid in the forms of fatty acid and few amino acids as well, they are entering in TCA cycle in the form of acetyl coenzyme A. Then that acetyl coenzyme A is forming different intermediate in TCA cycle, tricarboxylic acid cycle, forming different reducing equivalent, and at the end stage these reducing equivalent they undergo oxidative phosphorylation which forms ATP and regenerate those oxidized form of the equivalents. So, this is the concept of cellular respiration. Now, acetyl coenzyme A is the form by which actually pyruvate, which comes from glucose, or acetyl coenzyme A formed from different fatty acid or some of the amino acid, they enter TCA cycle. So, formation of acetyl coenzyme A is required. Now, the pyruvate we get from glycolysis, it forms acetyl coenzyme A with the help of the enzyme. Now, formation of pyruvate was in cytosol because glycolysis occurs in cytosol, but this enzyme pyruvate dehydrogenase complex is present in mitochondria, inner mitochondrial membrane. So, basically conversion of pyruvate to acetyl coenzyme A occurs in mitochondria. So, definitely acetyl coenzyme A must enter mitochondria. How it enters mitochondrial matrix? With pyruvate hydrogen symport. This is a transporter which actually carries acetyl coenzyme A inside mitochondrial matrix. Now, in mitochondrial matrix, the enzyme pyruvate dehydrogenase complex is present in the inner mitochondrial membrane. Now, this enzyme complex is a very interesting, is a very complex enzyme complex where there are three different enzyme activity. Basically, there are three different enzymes present. One is pyruvate dehydrogenase, enzyme 1. Then enzyme 2 is dihydrolipoyl transacetylase, and then dihydrolipoyl dehydrogenase. So, there are three different enzymes present in this enzyme complex. Not only that, there are 5 coenzymes or prosthetic groups. What are those? Thiamine pyrophosphate, flavin adenine dinucleotide, coenzyme A, nicotinamide adenine dinucleotide as well as lipoid. These are the 5 different prosthetic groups or coenzyme. Now, nutritionally this enzyme complex is also important because there are 4 vitamins which are required. Now, what are those vitamins? Thiamine, which is present in thiamine pyrophosphate, riboflavin present in FAD, flavin adenine dinucleotide, niacin present in NAD, and pantothenate present in coenzyme A. So, you can see in this enzyme complex there are 3 different enzymes present, 5 prosthetic groups of or coenzyme present, as well as 4 vitamins required for this enzyme's activity. So, how this conversion of pyruvate is happening with the help of pyruvate dehydrogenase complex. So, we can see there are sequential enzyme reaction by which pyruvate is converted to acetyl coenzyme A. This is the whole sequence of the reactions which is occurring inside the enzyme complex. Now, in the first step you can see pyruvate is converted to, pyruvate is actually releasing carbon dioxide. So, there is decarboxylation as well as the acetyl group is attached to TPP molecule. Now, this TPP after taking off the after taking up the acetyl group, it is converted to hydroxyethyl TPP. Then in the second reaction with the help of pyruvate dehydrogenase, this acetyl group is transferred to the core enzyme. What is the core enzyme? Dihydrolipoyl transacetylase. This is the core enzyme, and in this core enzyme there are 2 sulfhydryl group. This acetyl group is taken up by the oxidized lipoyl lysine group of core enzyme. So, this is the oxidized lipoyl lysine group of core enzyme, and it after taking the acetyl group from TPP, it forms the acyl lipoyl lysine. This is the acyl lipoyl lysine. Okay. Now, in the third reaction there is coenzyme A, which is actually taking up this acetyl group from the core enzyme, and it forms ultimately the acetyl coenzyme A, which regenerates reduced lipoyl lysine. So, this is the reduced lipoyl lysine. You can see the sulfhydryl groups are here reduced. So, all these are occurring inside the core enzyme, that is dihydrolipoyl transacetylase. Now, this reduced lipoyl lysine actually it gives off these hydrogen atoms to FAD present in the third enzyme, that is dihydrolipoyl dehydrogenase. FAD is reduced to form FADH2, and in the core enzyme once again the oxidized lipoyl lysine is formed. Once again you can see that from here the oxidized form is there. Now, this reduced FADH2 it gives up the hydride to generate NADH from NAD. So, once again the pyruvate dehydrogenase complex with its prosthetic groups with the help of the vitamins, they are ready for the next cycle of conversion of acetyl coenzyme A from pyruvate. So, at the end of the reaction what we are getting, that pyruvate releases carbon dioxide, decarboxylation, as well as it produces acetyl coenzyme A and also produces NADH, a reducing equivalent. So, now this acetyl coenzyme A enters the TCA cycle proper. Now, what happens in TCA cycle? Now, TCA cycle is tricarboxylic acid cycle, also known as citric acid cycle, also known as Krebs cycle, because definitely with the name of the discoverer Hans Krebs, it is named Krebs cycle. So, definitely it is a cyclical reaction or cyclical metabolic pathway where there are series of cyclical reaction inside mitochondria which oxidizes the acetyl residue of acetyl coenzyme A, and the reduced coenzyme reduced coenzyme, they undergo reoxidation in electron transport chain which forms ATP. So, this is the crux of TCA cycle. Now, TCA cycle is also the final common pathway for aerobic oxidation of carbohydrate, lipid as well as protein, because as we have already discussed, acetyl coenzyme A comes not only from glucose through pyruvate, but also from fatty acid by fatty acid oxidation and also from different amino acids. Then TCA cycle plays a central role in neoglucogenesis, lipogenesis and also interconversion of amino acids. We will discuss definitely in the later sessions. And with respect to TCA cycle, liver is the most important organism because this is the only tissue where all these metabolic pathways are happening. So, this is liver. Liver is the only tissue where all of these metabolic pathways are happening in a significant extent. Now, this is the TCA cycle. Let us see what is happening. So, you can see here acetyl coenzyme A, which is formed from pyruvate, is entering TCA cycle. It is combined with oxaloacetate, a 4-carbon molecule, to generate citrate. The name is citric acid cycle because now citric acid will undergo cyclical conversion to different molecules. So, there is formation of citrate with the help of the enzyme citrate synthase. With the help of the enzyme citrate synthase. Now, citrate synthase, why it is important? It is basically helping to form direct bond between carbon-carbon, the acetyl group of acetyl coenzyme A and the carbonyl group of oxaloacetate to form citrate. Now, citrate undergoes isomerization to form isocitrate. Now, this isomerization process is a 2-step reaction and the enzyme is aconitase. In the 2-step reaction, the enzyme is aconitase. First, citrate undergoes dehydration to form cis-aconitate. Cis-aconitate once again undergo rehydration to form isocitrate. So, this is the isomer of citrate. Now, this enzyme aconitase is inhibited by fluoroacetate. Now, what happens? Fluoroacetate, it forms fluoroacetyl coenzyme A. In the form of fluoroacetyl coenzyme A, it enters the TCA cycle, combines with oxaloacetate to form fluorocitrate. Now, this fluorocitrate inhibits aconitase. So, basically fluoroacetate undergoes modification to fluorocitrate which inhibits aconitase. Next, isocitrate undergoes oxidative decarboxylation to form alpha ketoglutarate. Again, this is a 2-step reaction. So, in this process, the first product is oxalosuccinate. From isocitrate, oxalosuccinate is formed by dehydrogenation with the help of the enzyme isocitrate dehydrogenase. As there is dehydrogenation, there is formation of NADH from NAD. Then there is decarboxylation again the enzyme is isocitrate dehydrogenase, decarboxylation. So, there is release of carbon dioxide, finally forming a 5 carbon compound alpha ketoglutarate. Next, this alpha ketoglutarate once again undergo oxidative decarboxylation, another step of oxidative decarboxylation with the help of the enzyme alpha ketoglutarate dehydrogenase. Now, alpha ketoglutarate dehydrogenase is a complex which is similar to pyruvate dehydrogenase complex. The whole enzyme reaction is just similar with pyruvate dehydrogenase complex. So, basically there are 3 different enzymes, there are 5 different prosthetic groups as well as 4 vitamins which are required for the enzyme activity. So, because that is decarboxylation reaction, there is release of carbon dioxide and also formation of NADH. Now, succinyl coenzyme A is converted to succinate. Now, this is one substrate level phosphorylation. What happens? In succinyl coenzyme A, there is a high energy thioester bond which finally helps in formation of ATP with the help of the enzyme succinate thiokinase. Remember, in all the kinases there is magnesium ion required as cofactor. So, there is formation of succinate which is a 4 carbon compound. Now, succinate undergoes different conversion to finally form oxaloacetate. What are those intermediates? From succinate there is formation of fumarate with the help of the enzyme succinate dehydrogenase. Now, succinate dehydrogenase is the enzyme which can be competitively inhibited by malonate, and in this dehydrogenation there is formation of FADH2 instead of NADH. Here in this dehydrogenation reaction there is formation of FADH2. Now, fumarate is converted to malate with addition of 1 molecule of water. You can see that here water is coming out, basically this is a reversible reaction, and from malate to fumarate there is release of water, but for formation of malate from fumarate there is addition of water, and the enzyme here is fumarase. Now, once again malate forms oxaloacetate by another dehydrogenation reaction with the help of malate dehydrogenase, and there is formation of NADH. So, as we have discussed that at the end of TCA cycle we are getting reducing equivalents, and those reducing equivalents are generating ATPs. So, let us see what are the net ATP production at the end of TCA cycle. So, in TCA cycle there are reducing equivalents like NADH. In these steps we are getting 3 NADH molecule. So, these are the 3 NADH molecule from where we are getting energy which is equivalent to, from each NADH molecule we are getting energy equivalent to 2.5 molecules of ATP. Fine. So, .5 molecules of ATP. Then there is another reducing equivalent FADH2, 1 molecules of FADH2. Now, FADH2 on oxidation it gives rise to energy which is equivalent to 1.5 molecules of ATP. Remember, previously the concept was 2 molecules of ATP are generated from FADH2, but the recent concept is that there is 1.5 molecules of ATP generated from FADH2. So, 1.5 ATP from FADH2, Then there is substrate level phosphorylation. So, in substrate level phosphorylation we are getting 1 ATP, substrate level phosphorylation we are getting 1 ATP. So, at the end of one TCA cycle we are getting 10 molecules of ATP. Now, remember that after glycolysis from one molecule of glucose we were getting 2 pyruvates, which is actually generating 2 molecules of acetyl coenzyme A. So, basically 2 molecules of acetyl coenzyme A undergo 2 cycles of TCA cycle. So, there is 20 ATP formation plus, if you do remember once again, from pyruvate to acetyl coenzyme A there were formation of 1 molecule of NADH. So, from 2 molecules of acetyl coenzyme A formation we are getting 2 molecules of NADH actually. So, these 2 molecules of NADH are giving rise to 5 molecules of ATP. So, at the end of glycolysis we were getting 7 molecules of ATP, glycolysis we were getting 7 molecules of ATP, and at the end of TCA cycle we were getting 25 molecules of ATP. So, the calculation which we can see that at the end of 1 molecule of glucose oxidized through aerobic glycolysis following TCA cycle to up to the end of electron transport chain, you can see there is 25 plus 7, that is 32 molecules of ATPs are generated. So, these are the key points discussed today, that is TCA cycle is occurring inside mitochondria. Pyruvate is converted to acetyl coenzyme A, that is also inside mitochondria with the help of pyruvate dehydrogenase complex. The resultant acetyl coenzyme A enters TCA cycle. In TCA cycle NADH, FADH2 and by following substrate level phosphorylation ATP are formed, and these electron carriers they finally enters electron transport chain and undergoes oxidative phosphorylation to form ATPs. So, this is the energy production through glycolysis and TCA cycle. Thank you.